Welcome back to our channel, The Exploring Medico. In this session, we will learn something about red blood cells, that is RBCs. So, we'll start with the characteristic features of RBCs. Functional morphology. The red blood cells, that is mature erythrocytes, form one of the important constituents of the cellular elements of the blood. We all know that it is the most important constituent of the blood in cellular elements. Each RBC, like any other cell, in the body is bounded by a cell membrane but is non-nucleated and lacks the usual cell organelles. In RBCs, there is no nucleus but it is bounded by a cell membrane, and it also does not have the cell organelles as in the usual cells. The cytoplasm of the RBC contains a special pigmented protein called the hemoglobin, which forms 90% of the weight of the erythrocytes. In RBCs, there is a special pigmented protein which we all know, that is hemoglobin, and it is in the form of 90% of the weight of RBCs. The red color of RBC and thus of the blood is due to presence of hemoglobin. Hemoglobin's main function is to provide red color to the hemoglobin. Then normal size, shape and counts of RBCs. Normal size, diameter of RBC is 7.2 micrometer and it ranges from 6.9 to 7.4 micrometer. Thickness in the periphery is 2 micrometer and in the center is 1 micrometer. The shape of RBC is biconcave, so in middle it is thin and in the periphery, that is at the border, it is thick. So in the periphery it is 2 micrometer while in the center it is 1 micrometer in thickness. Surface area of each RBC is about 120 to 140 micrometer square. Volume is about 80 micrometer cube and the range is from 78 to 86 micrometer cube. This we have to remember. Normal shape. The RBCs are circular biconcave discs. As we all know, the shape of RBC is biconcave and it is a disc shaped structure. The advantages of biconcave shape. This is the one of the IMP questions in RBCs. So we'll have to remember these advantages of biconcave shape. The first advantage is that it renders the red cells quite flexible so that they can pass through the capillaries whose minimum diameter is 3.5 micrometer. So due to this biconcave shape, the RBC becomes flexible and so it can pass from the capillary which have the size of around only 1 micrometer. So 1 micrometer to 3.5 micrometer. The biconcavity provides greater surface area as compared to volume, which allows considerable alterations in the cell volume. And the biconcave shape of RBC provides a larger surface area to it in the comparison to volume. So it contributes in the cell volume that the volume doesn't increases more, but in comparison to the volume the surface area is greater. Thus the RBC can withstand considerable changes of osmotic pressure. In this way the RBCs can resist hemolysis to certain extent when placed in the hypotonic solution. So RBCs can resist hemolysis. Hemolysis means breakdown of the cell to certain extent when placed in the hypotonic solution. Great surface area allows easy exchange of oxygen and carbon dioxide and rapid diffusion of other substances. So it is easy for the RBC to exchange oxygen and carbon dioxide due to this biconcave shape and larger surface area. As we can see here in the figure, the shape is biconcave. From middle it is thin, and from the periphery it is thick. And due to this, it can pass from the capillaries too, because of this biconcave shape. In the second diagram we can see that this diagram shows the flexibility of the RBC. Normal counts. Clinically, a count of 5 million per microliter is considered as 100%. The normal count of RBC is 5 million per microliter. Variations in size, shape, and counts of RBCs. So the variation in size is called anisocytosis. Then variation in shape is called poikilocytosis. And now we will discuss about variations in size. As I said, that it is called anisocytosis. Microcytosis, that is decrease in the size of RBCs, and macrocytosis, that is increase in the size of RBCs. Only two possibilities are there. The size can increase or decrease. So decrease in the size of RBC, and it is mainly due to iron deficiency anemia during prolonged force breathing and when osmotic pressure of the blood is increased. Now in macrocytosis, it is the opposite of microcytosis. That macrocytosis is seen in megaloblastic anemia. It is during muscular exercise and when osmotic pressure of the blood is decreased. In microcytosis, it is due to increase in the osmotic pressure, while in macrocytosis, it is due to decrease in osmotic pressure. Now, variations in shape. Variation in shape is called poikilocytosis. Abnormal shape of RBCs are spherocyte, then elliptocyte, sickle cell, and poikilocyte. Spherocyte means the shape becomes spherical instead of the biconcave. Then elliptocytes means the shape becomes elliptical. In sickle cell, uh, sickle cell means the shape of the RBCs become like a sickle. And sickle cell is seen in sickle cell anemia. Then poikilocytes. Variations in counts. Physiological increase in the RBC. Physiological polycythemia is seen in the following circumstances, that is age, sex, high altitude, excessive exercise, emotional conditions, and after the meals. We'll discuss it one by one. The first one is age. At birth, the RBC count is 6 to 7 million per microliter of blood. After about 10 days of birth, the count decreases due to destruction of cells. This is the cause of physiological jaundice of newborn. In infants, the RBC count is slightly more than adults. So, at birth, the RBC count is higher, that is 6 to 7 million per microliter. And after 10 days of birth, the jaundice occurs, physiological jaundice in newborn, due to which the RBC count decreases. In infants, the RBC count is slightly more than adults, as we have seen. Then sex. RBC count in the adult female averages 4.5 million per microliter, is lower than the adult males. Average is 5.5 million per microliter. So in females, the RBC count is less, and in males, the RBC count is slightly more than females. At high altitudes, individuals residing in high altitude areas above 10,000 feet from the sea level have high RBC count, that is 7 million per microliter, because of hypoxic stimulation of erythropoiesis. So people living in high altitudes have higher RBC count. Then excessive exercise. Mild hypoxia and spleen contraction causes temporary increase in the RBC count. So if we have done the excessive exercise, then mild hypoxia occurs, due to which the RBC count increases. Emotional conditions like anxiety are associated with the temporary increase in RBC count due to sympathetic stimulation. In emotional conditions like anxiety, we become anxious, and due to which RBC count increases temporarily. After meals, the RBC count is raised slightly. So we have seen six factors, that is age, sex, high altitude, excessive exercise, emotional conditions, and after meals. In age, we have seen that infants have higher RBC count than adults. In sex, we have seen that females have lower RBC count than males. At high altitudes, people have more RBC count. After excessive exercise, the RBC count increases slightly. In emotional conditions, even the RBC count increases slightly due to sympathetic stimulation, and after meals, the RBC count is raised slightly. Now, polycythemia. Polycythemia, or pathological increase in the RBC count above 7 million per microliter, is of two types, primary and secondary. There are two types. In primary polycythemia, or polycythemia vera, occurs in myeloproliferative disorder like malignancies of the bone marrow. In bone marrow cancer, malignant cancer, it is seen that there is increase in the RBC count. RBC count is persistently above 14 million per microliter and is always associated with high white blood cell count. So in this, WBC count also is higher, and RBC count is much more higher, that about 14 million per microliter. In secondary polycythemia, it occurs due to certain conditions producing a state of chronic hypoxia in the body, such as congenital heart disease and chronic respiratory disorders like emphysema. In congenital heart diseases and respiratory disorders, we can see there is secondary polycythemia, and they occur due to the chronic hypoxia created by these disorders. Physiological decrease in RBC count is seen in the following conditions. So decrease in the RBC count, that is at high barometric pressure, after sleep, and in pregnancy. In these three conditions, there is physiological decrease in the RBC count. And in anemia, there may occur marked reduction in the RBC count of the hemoglobin level, or both. This anemia is the pathological decrease in the RBC count, and it may be due to lower hemoglobin level, or both may occur in this anemia, that is decrease in RBC count as well as hemoglobin level. Now we will discuss about the functions of RBCs. There are many functions of RBCs. We'll discuss it one by one. The first function is role in carbon dioxide transport. Recall respiratory system, as illustrated schematically here with the unit of carbohydrate, produces about as many molecules of carbon dioxide CO2 as it consumes of oxygen O2. So HCOH plus O2 gives CO2 plus H2O. Thus the function of the circulatory system is as much about the transport of carbon dioxide as about the transport of oxygen. So the main function of RBCs is to transport carbon dioxide and the transport of oxygen. As stated elsewhere in this article, most of the carbon dioxide in the blood is in the form of bicarbonate ion. This we have to remember, and it is the main point that most of the carbon dioxide in blood is in the form of bicarbonate ion. The bicarbonate provides critical pH buffer. It is the one of the important buffer. Thus, unlike hemoglobin, oxygen for transport, there is a physiological advantage to not having a specific carbon dioxide transported molecule. Hemoglobin is used for the transportation of oxygen, but there is no mechanism for transport of carbon dioxide, and it is of much advantage to us. The second function is when red blood cells undergo shear stress in constricted vessels, they release ATP, which causes the vessel walls to relax and dilate so as to promote normal blood flow. So, when there is shear stress in RBCs in constricted vessels, they release ATP, which causes the vessel walls to relax. So when the shear stress is applied to the RBCs due to the constricted vessels, they release ATP, and due to which the vessels relax and dilate so as to promote normal blood flow. So it is much of advantage for us. The third function is that when their hemoglobin molecules are deoxygenated, RBCs release S-nitrosothiolols, which also act to dilate blood vessels, thus directing more blood to areas of the body depleted of oxygen. So, when there is deoxygenation in hemoglobin, RBCs release S-nitrosothiolols, which also act to dilate blood vessels. So, this chemical helps to dilate the blood vessels, thus directing more blood to areas of the body depleted of oxygen. Red blood cells can also synthesize nitric oxide enzymatically using L-arginine as substrate, as do endothelial cells. So RBCs synthesize nitric oxide. Then exposure of RBCs to physiological levels of shear stress activities, nitric oxide synthase and export of nitric oxide, which may contribute to the regulation of vascular tonus. The vascular tone, the tone of the vessels, is maintained by the nitric oxide synthase, which is produced by the RBCs when it is under the shear stress. The fifth function is RBCs can also produce hydrogen sulfide, a signaling gas that acts to relax vessel walls. It is believed that the cardioprotective effects of garlic are due to red blood cells converting its sulfur compounds into hydrogen sulfide. So, the cardioprotective effects of garlic is that it helps to convert sulfur compounds into hydrogen sulfide, and this hydrogen sulfide helps as a signaling gas to relax the blood vessels. Red blood cells also play a part in the body's immune response. When lysed by pathogens such as bacteria, their hemoglobin releases free radicals which break down the pathogen's cell wall and membrane, killing it. RBCs also play a part in immune response, as we all know, and when it is lysed by bacteria or other pathogens, the hemoglobin releases free radicals and it breaks down the pathogen's cell wall and membrane and thus kills them. Now we will see something about our red cell indices. The red cell indices defined below are calculated taking normal values of RBC count, that is 5 million per microliter, PCV, that is packed cell volume, is 45%, and hemoglobin level of 15 gram per deciliter. These are the normal values which is used in calculating the cell indices. The first one is mean corpuscular volume. It is the average volume of single RBC. We have to calculate that. It is calculated by dividing the PCV by red cell count. We can get the red cell count by counting the number of cells under the microscope. So the normal RBC count is 5 million per microliter. So PCV in 1000 ml of blood, that is PCV into 10 by RBC count per microliter. We have to count the RBC count and it is 5 million per microliter, as we have seen. The normal value is 5 million per microliter, and so it is 45 into 10 because PCV is 45%. So 45 into 10 divided by 5, it becomes 90 micrometer cube. So normal value of mean corpuscular volume, that is MCV, is 90 micrometer cube, and it ranges from 78 to 94 micrometer cube. When the MCV value is normal, it is referred to as normocytosis. Decrease in MCV occurs in microcytosis and increase in MCV occurs in macrocytosis. The next one is mean cell hemoglobin. Mean cell hemoglobin MCH is the average weight of the hemoglobin contained in each RBC. So how much amount of hemoglobin is present in RBC? It is measured by mean cell hemoglobin. It is calculated by dividing the amount of hemoglobin in 1 liter of blood by the red cell count in 1 liter of blood. So MCH is equals to hemoglobin gram per liter by RBC count per liter. So hemoglobin gram percentage into 10 by RBC count per microliter into 10 raised to 12, because it is in million. So we have to do that. So hemoglobin count, as we have seen earlier, that normal value is 15 gram per liter. So that we have to take 15 into 10 and divided by RBC count, which is 5 million per microliter. So 5 into 10 raised to 12. On dividing this, we'll get the answer 30 into 10 raised to minus 12 gram. That is 30 pg. It is the unit for 10 raised to minus 12. So normal value of MCH, that mean cell hemoglobin, is 30 and it ranges from 27 to 33 pg. It can vary in different humans as well as during different time of the same person. Increased values of MCH occur in spherocytosis and in the megaloblastic anemia. So MCH occurs in spherocytosis and megaloblastic anemia. Mean cell hemoglobin concentration. The MCH concentration, MCHC, refers to the amount of hemoglobin expressed as a percentage of the volume of RBC. So the amount of hemoglobin present in RBC is presented in the form of percentage. It is calculated by dividing the amount of hemoglobin in gram per deciliter by the volume of packed cells in 1000 ml of blood and then multiplying by 100. MCHC is equals to hemoglobin gram percentage by PCV by 100 ml into 100. So 15 by 45 into 100, which is 33.3 percentage. So normal value of MCHC is 33.3 percentage, and it ranges from 30 to 38%. RBCs with normal values of MCHC are called normochromic. So have mean cell hemoglobin concentration is normal, then it is called normochromic. In hypochromic, MCHC is less than normal, and in hyperchromic, it is the MCHC concentration is higher than the normal, and it is found rarely since the RBCs cannot hold hemoglobin beyond the saturation point. In hyperchromia, it becomes more than 38% of MCHC. So these three are important cell indices. We'll have to remember it, and it is one of the important questions. So now, in case of any doubts, you can ask in the comment section, and please like the video and subscribe our channel. Thank you.